Okay, well, I think we will get started. Um, really pleased to have as our speaker today Kevin Staley from the Mass General in Boston, Massachusetts. Before I introduce him, I want to acknowledge the support to underwrite this visit from Citizens United for Research in Epilepsy, otherwise known as CURE. This has really been a force in epilepsy research over the past decade or so, led by Susan Axelrod. You may have seen her on 60 Minutes and some other TV venues where she is really trying to combat the stigma of epilepsy and um, bring to the public's attention the importance of epilepsy research. So we're really grateful to their support, and we're grateful to Steve White from the Scientific Advisory Board of CURE, who's somewhere in our audience. Steve's from the University of Utah, where he heads the anticonvulsant screening program, and he's here just for this talk, so appreciate it. Our speaker today, Kevin Staley, let me tell you a little bit about him. He got his medical degree at University of University of California, San Diego. Also did his pediatrics residency and peds neurology fellowship there. Then went on to University of Colorado, where he rose through the ranks to a professor there in the pediatric neurology department. And then in 2006 was recruited to Mass General Hospital to be chief of the pediatric neurology division there, and currently holds the Rose and Joseph Kennedy, yes, those Kennedys, endowed chair in pediatric neurology. Kevin is really dedicated his investigational career in the study of GABA signaling and how it impacts epilepsy. He was really one of the first people to have the insight that because of disordered chloride homeostasis in immature animals and immature people, neonates that is, that our conventional GABAergic drugs to treat epilepsy and status might actually be counterproductive, and had the insight that if you use a simple loop diuretic to reverse that disordered gradient, that this might be that treating status in neonates might be more effective. And in fact, this has progressed to a clinical trial underway now. So a really sterling example of taking a fundamental idea all the way from the bench to a clinical trial. And I think I've interacted with Kevin over many years on committees and so forth, and I think everybody who knows him would agree that when you're talking science with Kevin, you're really in the no spin zone. He's refreshingly candid about his own science and that of others, so it's always a pleasure to have him. So Kevin, welcome.
Great. Thanks, Nick, for inviting me, and thanks to CURE also for this. This is their title slide. When I got it, it had Yvonne Soltesz up here as title. I thought maybe I'd change it to my name. I think that's good. But it's a wonderful program, and I appreciate being part of it. This is a movie of the latest calcium fluorophore called GCAMP6, and expressed with a virus and a subset of the neurons in hippocampal slice. It's an organotypic slice preparation that becomes spontaneously epileptic. Not directly relevant to the talk today, but I just brought it up as an example of the, we're in a golden age of physiology and pathophysiology in terms of the things I did in the first half of my career compared to the stuff I can do now with a couple of emails, including this sort of trick. And it's wonderful to be able to get insights into a seizure that I could never get with a single cell recording electrode for many, many years. So some things you really have to see the whole gestalt to figure it out, and we're really in that zone right now. So I have a few other examples of where a fluorophore can really make a big difference in terms of our insights. So I don't have any financial disclosures. I have a lot of intellectual disclosures. So Kyle Lillis made the movie you just saw. He's a great bioengineer who's done a bunch of things to make very high resolution, low light movies. That was actually a wide field microscope that we used to make that movie, and several very dedicated postdocs and graduate students who collected the data I'm going to show you today. So just to set this up, since it is Neurology Grand Rounds, I want to say that we're going to spend about 75% of the time going through some pretty interesting new physiology, and in the last 25% of the time talking about how that's going to affect the pathophysiology, some things that you see all the time in the neurology department. But to set it up, here's a baby, unfortunate baby with the neonatal herpes encephalitis. You can see that there are certainly seizure activity here, and there is diffusion changes on the scan and some swelling. And that went on to obviously encephalomalacia. This was not one of the patients who ended up with a recrudescence. I think Dalmau recently showed a high instance of NMDA receptor antibodies in this situation, but we won't talk about that today. What we want to focus on is this sort of a co-occurrence of edema, diffusion changes, seizures, and poor response to anticonvulsants that we see in the neurology department in a variety of settings. See it after trauma, see it after hypoxia, either global hypoxia in pediatrics or a stroke in the adult neuro ICU. And the question is, are all those things related? And the point that we're going to say is that these sort of pathophysiologies are linked in that we see increases in cellular salt water, and of course that reduces the diffusion. It can increase the ICP, but the chloride moiety of the salt water reverses the direction of GABA currents, and so instead of being inhibitory, they're excitatory, causing seizures. And of course, in that situation, our favorite anticonvulsants, propofol, pentobarbital, benzodiazepines, all just open the GABA channel longer, so they aren't going to do anything about a reverse GABA current, so they don't work very well. And so the idea then would be, I don't think it's a good idea to seize after brain injury. I think it's a second hit, and it can worsen the outcome. The only way to prove that is to have an effective therapy, which we don't have right now. And so the talk at the end is going to focus on some of these pathophysiologies and the clinical trial that Nick mentioned. But to get there, we've had a few recent insights that I wanted to share with you. As an electrophysiologist for many years, I have to say I'm as guilty as anybody else of thinking of neurons as sort of apples in a bucket of water, right? There's a neuron, and you patch on that guy, and around the neuron is an infinite extracellular space in which the chloride concentration is 136.5 millimolar, because that's what I put in the artificial cerebral spinal fluid. And I think that idea of the extracellular space probably needs a little bit of revision, but for right now, I just want to think about that the fraction of the brain that is occupied by extracellular space. And the way people have looked at that for a long time has been to diffuse some compound that you can track from one electrode, usually tetramethylammonium, and then record its diffusion at an ion-sensitive electrode nearby, and use this equation. Concentration is a function, obviously, of distance and time. Pretty hairy equation. And from that, you can get alpha, so the fraction of the extracellular space, the fraction of the brain that's occupied by extracellular space. And so this is a relatively smart rat. He has 21% of his brain is extracellular space. So, whatever it is, 79% is cellular elements. And on average, it's about 24% of your brain would be extracellular space. Of course, if something bad happens to you, we know that the diffusion coefficient drops, like we saw in that neonate with herpes encephalitis. And we see that the fraction of extracellular space drops accordingly here down to 6% of the total volume. And I guess the first thing I want to think about is 24% of your brain is not neurons, it's not glia, it's extracellular space. And why would we want to waste so much volume on that? We know that astrocytes can buffer potassium, can buffer all kinds of stuff. We don't really need to use 25% of our brain volume on extracellular space, and we pay an enormous advantage. I don't know if you ever tried to do this when you were a kid. I saw some movie, I think it was Magnificent Seven, and one of these gunfighters grabs a fly, flying through the room with his bare hands. I thought, I don't know, I was like 8 years old or something. I was like so impressed by that. So I ran out in the backyard and tried to grab flies, and of course, failed miserably. And now, after 50 years of training or whatever, I can say, I understand it, that I have much longer axon than the fly has, and so my reaction time is slower given the same conduction velocity. And so if you have 25% more space between your neurons, your ideas are going to come to you 25% more slowly, and that's assuming monosynaptic connections. Obviously, it's going to multiply as you start to think of polysynaptic circuits. If your axons are all 25% longer, it's going to take 25% longer for them to get to wherever they're going in terms of growth, so your development is going to be retarded by 25%. And obviously, you're going to have 25% fewer cognitive computing elements, so your cognitive capacity is going to be reduced appropriately. And finally, if any of you had babies or have been in a delivery room, you know about cephalopelvic disproportion. So the human pelvic canal barely fits the brain, and there are lots of babies and lots of women have died pretty horrible deaths because of cephalopelvic disproportion. The brain didn't actually quite fit through the birth canal. And so why would we, if we all say, well, there's cephalization, you know, the brain needs to get bigger, we need to think more, but why, if the space was that precious, would we waste 25% of it on the brain volume on empty space? And so I would say that the interesting thing is that, of course, the extracellular space isn't space. The way an electrophysiologist thinks about it, it's filled with this stuff called extracellular matrix, from which comes gelatin and jello and all kinds of other things, heparin. And this is made of complex glycoproteins. This is what the glycoprotein looks like under EM. This is a slightly enhanced artist edition of that. And this is a very enhanced artist edition. And basically, you have a hyaluronic acid backbone. You have these little red linking proteins. You have the longer core proteins. And the thing we're most interested in are the carbohydrates. So from each of these, we have these carbohydrate chains of variable length and variable sulfation. And here's an example of the various sequences of carbohydrates that you can have in the various glycosaminoglycans, all with different bizarre names, depending on which type of sugars that they have linked in these chains. But the thing we're going to focus on today is that there is no template to make these things. So it's completely untemplated in terms of how long each of these branches is. And there's absolutely no template for the amount of sulfation. So you can have 0, 1, or 2 sulfates. And of course, you have a carboxyl group on here also. So you can have very, very high negative charge density. In fact, there isn't anything more highly that has a higher negative charge density in the body. So these things are all freehand. They are linked to the intracellular space. So the extracellular matrix is connected to the cytoskeleton. And of course, it's connected to quite a bit of intracellular signaling cascades as well. If you don't buy the story I tell you today about the extracellular matrix modulating the reversal potential for GABA, you can also say, well, it could also be mediated by the changes that I'm going to show you in the extracellular matrix could be transmitted to these intracellular signaling cascades, and that's absolutely true. It's another alternate hypothesis. The extracellular matrix, as we know it now, we think about two things. We think, well, when you're developing, it helps guide axons to a particular place. And the other thought that we've had for the way the brain uses 24% of its volume is to modulate synaptic plasticity. So if you Google extracellular matrix, you're going to find a lot of stuff about perineural nets. Now, this drives me a little bit crazy. The extracellular matrix does not just exist around basket cells. This is a basket cell, but basket cells have a particular type of extracellular matrix that's pretty uniform and happens to be recognized by a plant, agglutinin from Wisteria. Let's go WFA here. But it's sort of like a plant antibody. It recognizes certain moieties and glycoproteins and will bind to it. And so it turns out that basket cells, you can make very pretty pictures of this perineural net, and I think you can see where the name comes from. But this stuff, as you'll see later, if you use an ionic stain instead of this sort of antibody stain, exists on all neurons. But the idea is, in the immature nervous system, the extracellular matrix is still developing. It's relatively low density. As time goes by, the density of glycosaminoglycans in the space goes up, and it's more difficult to force synaptic connections between neurons. That's been shown nicely in some time-lapse photography of spine formation in the young versus the old preparation. And in fact, Roger Chen, one one who developed a lot of the calcium fluorophores, has proposed that the extracellular matrix stores memory in the hole. So if you have a hole in the extracellular matrix, you allow a synaptic connection between neurons, and that's a form of memory. But I would say you can make a much richer form of memory, and there's that matrix all around the synapses, which will come up later when we talk about GABAergic synapses. But I would say that we can think of the extracellular matrix in terms of its information capacity. I'm not going to prove today that it's a source of, that it's a storage mechanism for memory, but I just want you to think about it for a minute. All the biology that we know about for DNA, RNA, and protein, those are all templated, right? You make a copy that your grandma had and your great-grandma had of DNA, and you make the RNA, which is in a way a copy of the DNA, and you make a protein from that. And we've been able to figure all this stuff out because we can amplify things. So we can do PCR or whatever to amplify the understand gene expression, because everything's an exact copy of everything else. We've got nowhere when it comes to glycosaminoglycan analysis, especially in the brain. And the reason is it's like studying snowflakes. Every one of these macromolecules is different than the last one. They're synthesized on the fly in the endoplasmic reticulum and the Golgi apparatus, and then exported to the extracellular space. We know nothing about the things that regulate the activity of the sulfotransferases that put those sulfates on, or what determines the length of the carbohydrates. So this is an interesting story. In 2007, you may remember that people who were loaded with heparin to anticoagulate the blood, it activates antithrombin 3, and is a sort of a clot dissolver, or inhibits clot formation. People were getting anaphylactic reactions, and nobody knew what was going on. It turns out that heparin is not made in a factory somewhere. It's actually the extracellular matrix from pig intestine, and it's collected from all over the world, probably places we don't want to think very hard about. You know, this is, and in 2007, people had to work very hard to figure out what the source of anaphylactic reaction was. The FDA was looking. There had been 81 deaths by 2008. And it turned out that they looked for a contaminant in the heparin sulfate, and if you Google it, you'll find out about contaminants. But what the contaminant was chondroitin sulfate, so one of these glycosaminoglycans, that had too many sulfates on it per sugar. And so the immune system can recognize the number of sulfates per sugar, and it's, to me, an example that of the storage, the information storage capacity of these molecules. So depending on how many sulfates, you can activate things like bradykinin and other parts of the clotting cascade that we don't normally want to target with the medical form of heparin, which is a fairly loosely defined gemisch of these glycosaminoglycans. In this particular spectra, these spectra are obviously not identical, and this is actually not the New England Journal paper. That's the one that describes the problem with oversulfated heparin. It's actually from the Brazilian Cardiology Journal. They were pretty pissed because once America and Europe found out what the problem was with heparin, they arranged to get better heparin and left all the poorly characterized unfractionated heparin to everybody else. And the Brazilian cardiologists were obviously not very happy with this. And this is a paper demonstrating what the nature of the problem was. So there is a lot of variability in the extracellular matrix components, and they turn over very slowly. They turn over about 10,000 times more slowly than an AMPA receptor. So AMPA receptor half-life in the postsynaptic membrane is about 10 minutes. And one of these glycosaminoglycans turns over about once a month. And that data is from a long time ago, probably the late 70s, so it probably needs to be revised, but I suspect that some elements of this would be probably even slower than that. So if I wanted to remember, you know, my birthday, I learned it when I was 5, and now I'm 55, so 50 years later, if I encoded that in AMPA receptor density, you know, sort of the thing we learn about long-term potentiation, there'd be 1 million turnovers of AMPA receptors between the time I learned my birthday and now. And so you would have to turn those over and replace them with no errors, over 1 million replacements. And I would say probably not a good way to remember your birthday. On the other hand, if you might do that shorthand, and then when you go to sleep and you transfer your memories to your cortex, I would simply say that extracellular matrix glycosaminoglycans would be an interesting way to store that memory for longer term, much lower turnover. And another way to look at the turnover is to look at the rate of symptoms that are acquired in these mucopolysaccharidoses, sort of an older term for glycosaminoglycans. And you can see that all these sulfated glycosaminoglycans need to be taken apart occasionally, and there are a whole host of pediatric storage diseases associated with enzyme deficiencies, all of which appear very slowly. So this, I think, reflecting the fact that the turnover rate is quite slow for these extracellular matrix components. So that's the extracellular matrix. The intracellular space, also chock full of anions. We all sort of know that because we know that the chloride concentration inside a neuron is only 5 or 10 millimolar. So there's got to be a lot of other stuff. There's 130 millimolar of potassium. So you have 120 millimolar or so of other stuff. These are beautiful pictures you can download from David Goodsell. The carbohydrates, proteins, nucleic acids, and the phosphate moieties associated with lipids are all negatively charged. There's also on tubulin a lot of glutamylation, so you have these long chains of glutamate that stick out from tubulin that are polymerized into microtubules in the neurons, and those are very reminiscent of the glycosaminoglycan strings that we were just talking about. Glutamate at intracellular pH is negatively charged. So we have intracellular macromolecules are negatively charged, phosphates on nucleic acids, all these amino acids that we were talking about, they're negatively charged at physiological pH. We have these polyanionic glycosaminoglycans on the outside, and so you can see that there's not very much room for chloride in this situation. And the argument that I want to make is that if neurons have a fixed volume and with this kind of complex geometry, I think it's fair to say that there's not a lot of room for the volume of the neurons to change a lot. If their volume is fixed, if you accept that, then the chloride concentration on either side of the membrane is really going to be determined by all the other stuff, by the immobile anionic macromolecules on either side of the membrane. And if that's the case, then the direction of GABA currents, which are mostly comprised of chloride ions, is going to be determined by the same thing, by the balance of intracellular and extracellular immobile negatively charged macromolecules. And so we're going to go into the Nernst potential a little bit. And so just a quick refresher, this is Walther Nernst. He came up with the third law of thermodynamics. And the idea is that the GABA reversal potential, we're just going to focus on chloride and skip bicarbonate right now because it's less permeable, that there's a concentration gradient across the membrane. And if you open the GABA channel, you'd say, okay, well, it looks like it's going to diffuse from the extracellular space to the intracellular space. And it will, but since it's negatively charged, you're going to have a gradual accumulation of negative charge quite rapidly before you actually change the concentration very much. And so that potential that you build up from unbalanced charge transfer is going to balance the diffusion tendency based on the concentration. So in that amount of negative charge that you build up, the potential is the Nernst potential or the reversal potential for the chloride. Another way to think of it then is that the GABA reversal potential is the voltage to which an open GABA channel is going to drive the membrane potential. So when it's open and chloride is diffusing, the membrane potential is going to move to this equilibrium that's set by the concentration balance on the inside and the outside. And the Nernst potential has this, Nernst equation has this kind of shape, so it's quite steep in the physiological range of a few millimolar chloride. It goes up from minus 100 to action potential threshold in just a few millimolar. So that means that if you change the chloride concentration, you're going to change the effect of GABA. It's pretty easy to do this experiment. I did it as an assistant professor, loaded a pyramidal cell with a high chloride pipette, and changed the GABA reversal potential so that when I stimulated an interneuron, instead of causing a hyperpolarizing IPSP, I got a depolarization in action potential. In that condition, if you then use our best anticonvulsant, kind of barbital, at an anesthetic concentration, all you're going to do is open the channel longer and get a longer string of action potential. So anesthetics are not going to work, anticonvulsants are not going to work if you reverse the if you alter the direction of chloride flow through the GABA receptor. Now that's a real dramatic example, but there's a whole range of things that happen between hyperpolarization and depolarization to the point of triggering an action potential. Lots of different ways that you can subtly alter the balance of inhibitory effects and excitatory effects depending on the reversal potential for the GABA A current. So at one end you have pure hyperpolarization. If you depolarize a little bit from there, you can have no hyperpolarization but shunting, so short circuiting of excitatory currents. As you go farther up, you start to remove inactivation of sodium currents, you start to trigger T-type low threshold calcium currents, remove the magnesium blocking the MDA receptor, etc. So more and more powerfully excitatory effects, but quite a range of things that can happen. And obviously in the GABA reversal potential is slightly positive to master memory potential. It's a pretty complex set of activities that can be triggered by GABA receptor activation. So clearly, I hope to make the point that the exact value of the reversal potential is pretty important for the GABA effect, and that exact value of the reversal potential, I'm going to argue, is due to this balance of macromolecules on either side. Now, that's not the conventional wisdom. Conventional wisdom, which we helped popularize, is that chloride, potassium, and sodium chloride cotransporters set E-GABA. So let me just tell you this is what you should fill in on a multiple choice test for the residents, and that is that the GABA reversal potential in an adult neuron is set by KCC2. So that's a potassium chloride cotransporter. It uses the energy in the potassium electrochemical gradient to move potassium and chloride out, so electroneutral, there's a positive and negative. Doesn't care what the membrane potential is, doesn't change the membrane potential, but the net effect is there's not very much chloride inside the neuron. So if you opened a channel that was only permeable to chloride, chloride will flow in. And that accumulation of negative charge would hyperpolarize the neuron. And so that's something good. If you wanted to stop seizures, you'd do more of that. You'd open this channel longer with barbiturates or propofol or something else. With an immature nervous system, GABA is thought to be excitatory. There's a different transporter, so KCC2 is not expressed, and KCC1 is expressed instead. It uses the energy in the sodium gradient to bring sodium, potassium, and two chlorides in. The net effect is too much chloride on the inside. And so when you open the same channel, chloride is going to flow out, and the loss of negative charge depolarizes the neuron. This is the transporter that we block with bumetanide. So this looks like a great scheme, and it's something that you can kind of remember pretty easily and attractive in that way. And of course, like everything else, it's simple and easy. There are a bunch of problems with it. One is these are both equilibrium transporters, and you can never find a neuron where the chloride concentration matches the reversal potential for equilibrium concentration for the transporter. So this is real easy. There's only potassium and chloride. You know the potassium on the inside, you set the potassium on the outside, you set the chloride on the outside. That means you've determined the chloride on the inside. It never works that way. And the same is true for NKCC1, though. It's a little bit more complicated to go through. When you block the transporters, the chloride never changes very much. So if these guys are really working to set the chloride transport, then you think when you block them, you'd see a big change. And in healthy neurons, not unhealthy neurons, but healthy neurons, that's not true. The transporters often run backwards, so they don't always go in this direction. They're equilibrium, and they can go in either way. And the chloride varies widely in different parts of the cytoplasm, which doesn't make sense. If you pump chloride in over here and out over here, you're just going to end up with a circuit of chloride flowing like this and just waste a bunch of energy. So there has to be some other way to sort this out. So let me go through the ways that we think are going on. We use this chloride-sensitive fluorophore called chlumillion to study the studies I show you in the next couple of slides. Chlumillion was made by George Augustine and Thomas Kuhner. It's a pH and chloride-sensitive combination of yellow fluorescent protein and cyan fluorescent protein. Yellow fluorescent protein is pretty easy to quench the emission with chloride, with oxygen, with protons. And here's just the pH sensitivity, so we have to pay attention to that and sometimes measure the pH in some of the experiments I'm showing you. But overall, in the physiological chloride range, is pretty modest. Cyan, on the other hand, isn't quenched by anything. The only problem we have with cyan is that it's scattered a lot more by tissue. So if we try to do something tricky, like look at the pyramidal cell layer, the neocortex in a living animal, we have to recognize that we've lost a lot more cyan fluorescence than yellow fluorescence. It makes it pretty hard to calibrate the dye. The other thing about the dye is that it has a relatively shallow curve in terms of the change in yellow fluorescence versus the change in chloride. So 1% change in yellow fluorescence is about 3 millimolar shift in chloride, which is a lot. So there's a new dye now, superchlormelene, and we're replicating a lot of these studies with the dye that has a much steeper chloride quench curve. So I'm going to show some slides like this where we color the neurons according to the chloride concentration. So here's the calibration bar. Now this is millimolar chloride, sorry, here, and this is the chloride reversal potential. I think I got those. Sorry, this is the millimoles chloride here. Sorry, the labels off. This is the corresponding chloride reversal potential. And so these pseudo colors mean that the dark blue guys are in the physiological range, and the hotter the color, the higher the chloride. When we made a overall distribution of these chloride concentrations, we found a pretty normal distribution, very broad, much broader than we expected. And the distribution was not at the NKCC1 reversal concentration, nor was it at the KCC2. In fact, almost no one was at either of those. They were all in between. This wasn't an artifact of the dye. We see similar. So that's a good point. So this picture here is not very good, but the sort of bean thing is supposed to be a whole hippocampus. So we shelled out a whole hippocampus, so it didn't do any slicing, and imaged this. I'll show some effects of slicing in a couple of slides. So you look at the distribution in perforated patch recordings, which is a non-invasive way to look at the chloride using a cation permeable ionophore between the electrode and the neuron. You see the same distribution of chlorides. If you look at cell attached, so you can also measure the distance between resting membrane potential and the GABA reversal potential using two cell attached patches that look at channel openings to GABA and NMDA. And you can't get the absolute value of the reversal potential, but you can get the distance between the GABA reversal potential and resting membrane potential. And the point of this is that that value never matches the equilibrium point for KCC2 or NKCC1. So using fluorometric methods or perforated patch methods or cell-attached patch methods, nobody can find a situation where the chloride reversal potential or the chloride concentration matches the equilibrium potentials for either of these currents transport. And you could say, well, that's because they're both running at the same time. This is push-pull, and you have chloride going in one way and coming out the other way, and it's a nice way to set up exactly where you want the chloride to be. And that might be true if you had nanomolar concentrations of chloride, but we have millimolar concentrations, and these transporters move millimoles per liter per second. So you basically evaporate all the ATP in your body if you let these two guys run against each other like this. And so to show that, what we're going to do in the next experiments is block this guy, or block this, and then block this guy, and we're going to expect the chloride to move this way or that way as we block one of these transporters or the other. Here's an example of that experiment. This focus on here. Here's the initial chloride concentration. Here's the final chloride concentration. Black is after we blocked NKCC1 with bumetanide, and then we sequentially and specifically block KCC2 with this BU compound that Eric Delpierre made that's a specific KCC2 antagonist. And what we see is, obviously, the best predictor of the final chloride concentration is the initial chloride concentration. This is what it would look like if the transporters were determining the chloride concentration. When we block both of them, chloride would be passively distributed, where E chloride equals resting membrane potential, and it wouldn't matter what the initial chloride is. So all the points should lie on this line. Instead, we see that they aren't anywhere near that. We could do the experiment in an adult, get the same thing. We could reverse the sequence, so block KCC2 first and then KCC1 second. Doesn't make any difference. If we add up how much the chloride changed when we blocked the first transporter, and then how much it changed when we blocked the second transporter, they're in different directions, but if we took the absolute value, it should be about 15 or 20 millimolar chloride difference. What we see instead is distribution around 0. So there is no real effect of blocking these transporters. So we feel like these transporters are not acting against each other. Energetically, it doesn't make sense, and there's no pharmacological evidence for that either. The transporters don't run in the direction they're supposed to in most of these cells. So this is the same data type I showed you on the previous slide, but we plotted it a little bit different. The initial chloride versus the changing chloride. In the previous slide, this was the final chloride. And so the yellow, this is the canonical direction. So if we take, in this case, NKCC1, a chloride accumulator, you expect when you blocked it, that chloride concentration would decrease, and instead we see about half the time it increases. In the adult chloride exporter, when we block it, chloride should go up, but most of the time it goes down. So there's clearly these guys are fairly large effects here. So chloride's moving through these transporters, but the transporters aren't what's determining the reversal potential. The other problem that we had was this subcellular variance in chloride. So the other problem with chloride transport determining the chloride concentration is that the chloride concentration can differ in different parts of a neuron. So it could be high in one area, low in another. This was first pointed out by Spatix et al. In Science in 2006. They were into the effects of different interneurons on pyramidal cells, and they noticed that exoaxonic cells that inhibit the axon initial segment had a reversal potential of about minus 55 millivolts, whereas basket cells that inhibit the soma have a reversal potential of about minus 75 millivolts. So 20 millivolt difference from axon to soma. Hard to explain that based on pumps, but pretty easy to explain if you, so we replicated that data basically. It showed that it persists, that soma axon gradient persists after we block chloride transport with bumetanide or with, I think in this case we use furosemide in the adult. So that we see some changes in the chloride concentrations, but the gradient from soma to axon, which goes this way in the young animal and in the opposite direction in the adult, is not changed by blocking the transporters. So then what's going on? What we would say is the reason that in a healthy neuron there's no effect of the transport inhibition is because what's really determining the chloride concentration, which I think makes I think is reasonable when you think about it, is there's other stuff inside the neuron besides chloride. And blocking the transporter doesn't remove that other stuff. It still only leaves a certain amount of room in the neuron for chloride. If you have a lot of other stuff, a lot of immobile anions, then there's very little room for chloride to be distributed. If you don't have very many immobile anions, then there's quite a bit more room for chloride to be distributed. And we measure the mean when we do electrophysiology or these fluorometric measures. So there's some straightforward predictions from this. If this is a true idea about what's going on, then we expect that chloride should vary inversely with the amount of other stuff. We can test that by looking at the correlation between the other anions and chloride. We can change the amount of other anions and see if that changes chloride. And similarly, we would expect in the extracellular matrix that if there was a lot of polysulfated glycosaminoglycans, then there shouldn't be very much chloride on the outside. And KCC2 links these two things together, intracellular and extracellular chloride, so that the intracellular chloride should vary inversely with the extracellular anion concentration. So we'll test those two things. So here's the first test. This is a test for all the intracellular fixed anions, most of which we can't stain. I don't know how to stain for amino acids that are negatively charged to physiological pH, but I can stain for nucleic acids, which I know each base has a phosphate associated with it, and there are lots of stains for that. This is Cyto64, which bonds covalently to nucleic acids. And so we looked at the intensity of Cyto64 staining versus chloride staining in chlamydian mice that were subsequently stained with Cyto64. And we got a reasonable correlation coefficient of about 0.3, which makes sense because about a third of the immobile intracellular anions are made out of these phosphate groups. So we didn't expect to get a much higher correlation coefficient than this. The other way to do this then would be to change the intracellular anion concentration and see if we can change the chloride concentration. This idea was breached first by Reams in a very sort of controversial neurochem paper in 2009, where they argued that beta-hydroxybutyrate is what's in rat's milk. That's what the babies feed on. And the reason why GABA isn't excitatory in all these brain slice experiments that people do is because we don't put any beta hydroxy, we don't put any ketone bodies in our artificial cerebral spinal fluid. We force them to eat sugar instead. I don't think that's what's going on. I think if you take an organic acid that has a transporter, it's going to be passively distributed according to the resting membrane potential, and at a minus 75 millivolt transmembrane potential, you get about for monovalent anion, 20 millimole on the outside. Millimole on the outside will put about 1 millimolar on the inside. That's easy to show with something like sodium gluconate. So we can show that we decrease the intracellular chloride by about a millimolar with gluconate. Lactate doesn't change very much, but this transport is already saturated under physiological conditions, and pyruvate we can get a little bit of a change too. So when we change the intracellular anions, we change the intracellular chloride in the opposite direction. There's a better way to do this experiment, which is just to take decades of chloride recordings from whole cell and sharp electrodes, and people have shown since Hodgkin and Fat back in the 50s that if you put an anion besides chloride in your recording pipette, you're going to change the chloride concentration in the neuron. This is just an experiment that we did a long time ago, showing an inverse correlation between the amount of gluconate in the electrode and the chloride reversal potential measured at the GABA receptor. So a little bit of variation in that experiment I showed you before, where chloride was excitatory. So the other half of this is what happens when we change the extracellular matrix, where I've made all those wild speculations about memory. So if we change the extracellular matrix, those fixed anions, what happens to chloride? So here's the idea. We're going to take these sulfates and move them. We're going to chop them off. And what's going to happen then is the chloride's going to diffuse into that space. And we're going to end up with a higher chloride concentration on the inside, so raising the chloride concentration potential. And now KCC2 needs to balance this ratio, so we're going to increase the chloride on the inside from 1 millimolar to 2 millimolar and even that out. So KCC2 is going to reflect the change in the concentration on the outside, which I don't have a way to measure, by changing the chloride on the inside. And so that's easy to show. Here's the correlation between the extracellular matrix first and the intracellular anion. So remember we talked about perineural nuts before. This is an ionic stain, alcium blue. It binds to glycosaminoglycans, binds to anything actually anionic. But at pH 0.5, the only thing that's still anionic that hasn't been protonated is the sulfate groups on the glycosaminoglycans. So pH 0.5, this is a pretty specific stain of glycosaminoglycans. And what we can see is around every pyramidal cell, we see one of these perineural nets. So if we look at alcium blue staining in the hippocampus and the pyramidal cell layer, and we correlate it with the chloride concentration, so we did this experiment first, and then we stained alcium blue, we get the expected inverse correlation that we would see. Now, we didn't look at the dentate, which is interesting because there's a little bit more alcium blue stain there, but at this age, there wasn't any chlomelain expressed. So hopefully in the superchlomelain mice will be able to look at the dentate. So if we decrease the extracellular matrix, well, we showed that in the cartoon before, but basically we're going to take these glycosaminoglycans and chop them off, leaving more room for chloride to flow in. Then we're going to see what happens to the chloride concentration on the inside of the neuron. What we see is when we use chondroitinase ABC, so this is something that the spinal cord injury people use a lot to remove the extracellular, partly dissolved the extracellular matrix. This removes the chondroitin sulfate. We see a pretty dramatic increase in the chloride concentration in these neurons. Basically doubles. And the idea then is that if we remove some of these, this is intracellular versus extracellular components. If we shrink the sulfate moiety, we're going to get less sulfate, more chloride on the inside, and then KCC2 is going to balance that by increasing the chloride on the inside. So the product of KCl remains the same on both sides. So this is interesting. That means that you don't have to switch from NKCC1 to KCC2 during development. in order to change your chloride, all you really need to do is increase the complexity of the extracellular matrix and the intracellular anions. And that's already been shown that, for example, the protein concentration inside neurons increases pretty dramatically with development over time. It's been shown repeatedly that that's also true in the adult extracellular matrix. And so what does that mean? That means that not only is GABA determined passively by these macromolecules, but it means that the GABA reversal potential can vary at every GABA receptor. And we don't normally think of it that way. We normally patch on a single neuron, and we activate the GABA receptors globally or with a very large macro electrode, and we expect, and we get some sort of average for the whole neuron. But in fact, it's often a little bit of polyphasic changes. And what we see when we look in with chlomelion, and we're just now repeating these experiments with the more sensitive superchlomelion dye, we see these microdomains of chloride in the dendrites, suggesting that chloride can change depending on the local concentration of intracellular anions. Then we develop intracellular Donin equilibria as well as Donin equilibria across the membrane. And so you can imagine that if we have a section of dendrite here, and the A's are anions and the chlorides are chloride, that you can have stretches of dendrite with a lot of anions. That's going to have a very negative GABA reversal potential. There's not very much chloride here. In places with progressively more chloride with progressively more positive chloride reversal potentials. So that chloride will flow in this area. So this GABA receptor is hyperpolarized and inhibitory. This one, the chloride reversal potential is right at resting memory potential. So it's there's shunting inhibition here. And here it's frankly depolarized. And these three receptors could conceivably lie on the same GABA receptor. And if we changed either the intracellular or the extracellular anion concentration, we could change the reversal direction and the amplitude of these currents. So that's what we say here. I don't think that KCC2 and NKCC1 set the chloride concentration. I can't find very much evidence for that. I think the chloride instead varies as a function of these extracellular macromolecules, and that explains the subcellular developmental variance in chloride. And if you accept that, then you can also accept that these slowly changing macromolecules could subserve an analog form of memory. Not that they do, but they could. And certainly they have the right turnover for that sort of thing. Changes in the intracellular anions change the GABA reversal, and the extracellular changes have a bigger effect on the conductance. But the KCC2 and NKCC1 are important, and they do maintain chloride in a healthy neuron. And what I want to show now in the last couple of minutes is what happens after a brain injury. So we started with this slide. If you think about this delicate balance of intracellular and extracellular macromolecules, if you damage it in some way, like a head trauma, for example, or an infection or hypoxic injury or prolonged seizures, you can change the balance of intracellular and extracellular matrix. You can end up with cytotoxic edema, so a shift of salt water from the inside to the outside. That's going to cause the diffusion changes that we've seen already. Could cause ICP changes if it's widespread enough. But the thing that we worry about is, in terms of seizures, is if the chloride moiety of that salt water gets inside the neuron, it's going to change the reversal potential for GABA currents and lead to seizures. And that's why we often see in the ICU a combination of ICP problems, diffusional changes on MRI, seizures, and poor response to GABAergic anticonvulsants. So it's pretty easy to show that trauma causes chloride increases. We got in trouble a couple of years ago with this paper in January Psi, where we said that in acute slice, it's a nice trauma model because we basically take a razor blade, a vibrating razor blade, and cut right across this surface. And we go down 400 microns and we make another cut across the surface. It causes diffuse axonal injury and all kinds of what we would call a shear injury in the radiology suite. When we look at those neurons, what we see, of course, they're pseudocolored here, very swollen, high chloride neurons with a lot of salt water brought into them. And that's just shown here. If we look at repeated seizures, we see the same thing. So we can take a preparation, in this case, a whole hippocampal slice. So again, not traumatized with the razor blade, subjected to low magnesium, which induces seizures, and with each seizure, the chloride goes up and up and up, as shown here. That can be blocked with bumetanide. So this is where bumetanide may come in handy after injury. It inhibits some of the backward flux through NKCC1 and the rate of chloride accumulation. So if that were true, then we'd expect that a GABAergic anticonvulsant would work great after one seizure, poorly after many seizures because there's been chloride accumulation and now chloride is flowing the wrong way through the GABA channel. And that just shows that with each seizure, the phenobarbital efficacy decreases. The other implication is that we'd expect that if we combined the two drugs, if we inhibited the chloride influx through NKCC1 and we used phenobarbital to open the GABA channel better, that should work as a better anticonvulsant after multiple seizures, and it does. This is just the group mean data showing that phenobarbital plus bumetanide works a lot better than either agent alone. So the fundamental idea, of course, is we want better outcomes. We still don't know in 2014 if neonatal seizures are a biomarker for something bad that happened to your brain, hypoxic injury, stroke, hemorrhage, herpes infection, or if the seizures are actually making the situation worse. Because we don't have a way to treat and block the seizures, we can't really test this outcome very actively. And what we have instead is a bunch of agents that don't work. Here's a pretty famous study of phenobarbital versus dilantin. The idea being that this is the natural history of neonatal seizures. And if you treat neonatal seizures here where the incidence is increasing, there's very poor efficacy, about 24%. If you treat neonatal seizures when the frequency is decreasing, everything works. Bible dust would work in this case. And so they work much better. And most people quote this overall efficacy of 58%, which is the average of all these, but I would say this is the only one that matters, 24%. Here's an example of this, Keppra. People say works great. 32% respond in an hour, 64% respond in 24 hours, 86 respond in 48 hours. Oh, Staley, neonatal seizures are cured. We'll just use Keppra. It's a very benign drug. It's available IV. But this is exactly the same as the natural history of untreated neonatal seizures. In this case, neonatal seizures treated with phenobarbital, which doesn't work at all. So if you superimpose these two studies, you see that Keppra hasn't really altered the natural history of neonatal seizures at all. So what we want to do then is we've set up this Boston bumetanide trial. We think that what happens is that we get an imbalance, injury after hypoxic injury of the sulfates on the outside and the immobile anions on the inside, and that water fluxes back and forth to the transporters, salt water fluxes back and forth through those transporters. And what we really need to do is block those things to prevent chloride accumulation. So we've taken neonates that are at risk or have obvious evidence of seizures and treated them with phenobarbital, which is, of course, is clinical protocol all around the world right now. If they have further seizures, then we randomize them to either more phenobarbital, up to 40 per kilo, or we give them phenobarbital plus bumetanide. So nobody gets salt water. You either get phenobarb or phenobarbital plus bumetanide. And so the question is, what are the pharmacokinetics? And do these drugs, this is the maximum diuretic dose in neonates, when we're stepping from 0.1 to 0.2 to 0.3 to see if this dose will impact seizures and whether it's toxic at all. This study was also initiated in Europe as the NEMO trial, and it was a dose-finding trial without a control group, which turned out, it was a Bayesian thing, so they were going to randomly allocate people to different doses, and whoever did better, they would allocate more people that way. So the idea is that you rapidly find the correct dose. It doesn't correct at all for bad things happening. They had two neonates who developed unilateral hearing loss, one in the lowest concentration group, one in the highest concentration group, so they just stopped the study. They weren't super enthusiastic about it anyway. So we've continued it in Boston. We haven't had anybody have hearing loss, and we will be unblinding the first two, I hope we're going to be unblinding the first two flights of this, so 0.1, 0.2 per kilo later this summer. We have the last two babies to enroll. So lots of collaborators. This, I think, as I said in the beginning with, a neat movie of seizure onset in organotypic sites, this is a great time in neuroscience and certainly in understanding the pathophysiology of a lot of diseases. We have tremendous tools at our disposal to look at things that have stumped us for a long time. And I think it's a fantastic time to be involved in neuroscience research. Thank you.
So
Sure, right.
nice work. But based on your talk, perhaps the midnight probably would not work if the other ions are more important.
Yeah, so that's a great question. So you're right. And if the neonates were healthy, I wouldn't expect bumetanide to do anything. And that's what we show. We show a bunch of movement with a mean of 0. So we wouldn't expect anything at all. I think what happens in injury, we have a different story. So when I show the slices with the swollen neurons near the cut slice, the same thing's been shown for hypoxic injury. I didn't show that here, but Augustine showed that in 2006, and we've verified that also. So with an injury, you get a derangement. I'm not sure where the derangement comes from. I think it starts off with a little bit of necrosis in release of hydrolases from the lysosomes inside some necrotic cells. They disrupt the extracellular matrix, changes the chloride concentration, changes the extracellular chloride concentration, and causes influx mediated by the transporters into other neurons. And that, one thing about edema that doesn't ever make any sense to me is that it takes 3 to 5 days to peak. So there's some kind of gradual thing happening. And I would say dissolution of the extracellular matrix by these enzymes that are released from the necrotic neurons is one possibility anyway. So you're right. In physiological conditions, the net effect of the transport inhibition is basically 0. But in pathophysiological conditions, it works better. It doesn't work very well for trauma. So we see a modest change with diuretics and trauma. It works great for increases in chloride caused by repeated seizures or by hypoxia. I don't really have an explanation for why trauma is so much different than the other two.
Also, are there clues from some of the other disorders that we see in PPF? So the disorders of glycosylation, often epilepsy is a very presenting symptom or is also untreatable as well.
Correct.
And the specific macromolecules and glycoproteins that are modified in those disorders may provide the clues which are the critical molecules that are important for both in the extracellular space as well as intracellular.
That's a great point. And congenital disorders of glycosylation used to be my least favorite disease because it was just so poorly understood by me. And now it's one of my most favorite diseases for exactly that reason, because maybe that's the way to explain it. Now, it turns out there's all different kinds of glycosylation, some of which happen in the brain, some of which happen elsewhere, and some of the congenital disorders may not happen in the brain, but some of them do. And certainly the intellectual disability that's so commonly associated with that, sometimes you go, well, the extracellular matrix isn't right, the brain's not going to build itself right. True enough, but a lot of times the brain's morphologically pretty normal. Too bad Bill's thumb's not still here, and yet those kids are profoundly retarded. So I absolutely agree. I think that you can have problems with the sulfotransferases, or you could have problems with building those carbohydrate strings, or breaking them down. And in pediatric neurology, we mostly think about breakdown. We're just starting to think about the build up, but I think they're both really critical. Sure.
Anything particular about bumetanide as opposed to the other loop diuretics?
Yeah, so you could use furosemide if you want. Furosemide blocks KCC2 and NKCC1. There's not a lot in the developing nervous system. There's not a lot of KCC2 around. There's some. And it might work better than bumetanide, particularly since we've shown that we get bidirectional transfer of both. When we set the trial up, we really thought that KCC2 was the good guy who was pumping chloride out and making GABA inhibitory, and NKCC1 was the bad guy bringing chloride in. So we used a NKCC1 specific transport antagonist. But if you look in the literature, people have reported that furosemide, for example, blocks, when people get furosemide before neurosurgery for epilepsy surgery to shrink the brain down a little bit and prevent swelling, that they actually inhibit interictal spikes. So I think that you could argue that furosemide would be better. Like I said, there's more NKCC1 in the developing brain than KCC2, so I think that bumetanide should work well. I don't know which one crosses the blood-brain barrier better. And that's the one big unknown in our study is how well that's going to work. And it may turn out that if bumetanide doesn't work, that furosemide may. On the other hand, you really only get one chance to do a clinical trial. So if this doesn't work, I doubt we'll get another chance with furosemide. Okay, well, thank you.